Hello and welcome to this IChemE Global Awards webinar. My name is Matt Stalker and I'm your host for today's presentation. The IChemE Global Awards celebrate chemical, process and biochemical engineering excellence and are widely recognised as the world's most prestigious chemical engineering awards. Today we'll be announcing the winner of the IChemE 2020 Pharma Award sponsored by PM Group. This award recognises the best project, process or product to demonstrate chemical engineering excellence in the pharmaceutical sector. And joining us now is PM Group's Regional Development Director, Peter Farrelly, and Technical Director and Deputy Pharmaceutical Sector Director, Austin Locke.
Hello, everybody, and good morning, or good afternoon, or good evening, depending on where you're joining us from. We're especially pleased to sponsor the Pharma Award as part of the IChemE Global Awards 2020. In these very challenging times, it has never been more important to support the continued development and promotion of excellence in our industry. As an international project delivery specialist, we in PM Group are very comfortable with complexity and innovation. In fact, these are things that are at the heart of all of our projects. For almost 50 years, we've been helping our multinational clients to design and deliver their latest facility developments across the world. The skills and expertise to do that successfully have to be nurtured and built over the years. And for us, that starts with our graduate training scheme, a scheme that's recognised and supported by IChemE. And then those skills and that expertise is built up over the years through not only on-the-job learning, but also by continuous professional development activities. The IChemE Global Awards are helping to inspire greater investment in this kind of expertise, leading to greater innovation and, we hope, a more sustainable future for all of us. I'm pleased to now hand over to my colleague, Austin Locke, our Technical Director and Pharma Sector Director, who will talk to you more about innovation and how it's helping us and our clients and industry alike. I wish the best of luck to all the finalists today, and I hope you enjoy the presentations. Thank you.
Thank you, Peter. I'd just like to check before I start. Can everyone see the presentation?
Yes, we can.
Okay, brilliant. Okay, so thank you, Peter, and hello, everyone. First of all, I'd like to start by giving my congratulations to all the finalists for their excellent achievement in demonstrating pharma innovation. And with that, I want to spend the next few minutes just talking about PM Group's approach to pharma innovation, because like the finalists companies represented today, PM Group drives pharma innovation. So within PM Group, there's 4 routes of innovation. We have the Innovation in Action scheme, which is an annual awards program which encourages everyone from the company to promote concept innovations across a wide range of areas. And then we have innovations generated within the IS and digitization groups, innovations generated within the project teams themselves and departments. And then we have innovations generated within the tech team. And it's the tech team I want to focus on today. So this is the tech team. It's comprised of approximately 50 multi-sectoral, multidiscipline specialists from across PM Group's 15 offices worldwide. When I talk about the tech team to our clients, I refer to it as a community. We meet every other month by WebEx, and we meet face-to-face annually in normal years anyway, at least. And it has 3 main objectives. It provides a forum for ideation. It undertakes internally funded development projects, and it provides community for knowledge exchange and expertise. So what does this bring to PM Group? Well, apart from delivering on advancements in facility design and technologies, it also provides PM with a significant reputation as being an innovative company. One thing to highlight about the tech team is it's very much outward focusing. We collaborate with a number of companies in our innovation projects, and these are a list of companies we're currently engaged with, and they can be broadly categorized into four main groups. We have academia, industry bodies such as the IChemE, client organizations, and suppliers. And I can't emphasize enough how important these collaborations are to PM Group innovations. With regard to activities, the tech team undertakes a number of, as I say, internally funded projects, which can be broadly categorized into three main groups: Industry 4.0, single-use facility design, and facility of the future. And just to give you a flavor of the type of projects that we're engaged with, in Industry 4.0, we are engaged in a number of robotic associated projects. So we have Project Marvin, where we're working with a number of companies, Novartis, Lonza, UCD, Nibert, in the development of an autonomous environmental robot, which collects clean room environmental samples and interfaces with software, which not only manages the activities of the robot, also performs data analysis. In Project Khan, we have worked with the Technical University Dublin to undertake a scientific study to determine the effectiveness of IPA wipes surface sanitization in airlocks. And we're now looking for additional partners to undertake a proof of concept using UV sanitization on robotic arms, again to determine the effectiveness of surface sanitization in airlocks. And in Project Arthur, we are providing technical expertise to provide a standard-based approach to the integration of analytical equipment with a QC stack robot. With regards to single-use facility design, we have many innovations associated with facility layout, single-use component management, and logistics. However, briefly today, I just want to focus on our single-use standardization approach. It's an industry-leading approach called SDD. Single-use standardization will bring many advantages to client organizations, and we've been engaged in this activity for over 6 years now. It's involved tens of biopharma companies and a number of single-use suppliers. And basically, we've generated a single-use standards part of about 400 parts. And we have now agreements in place with a number of single-use suppliers to supply those parts. And we've also got a number of our clients now adopting SDD and obtaining the benefits that provides. And from that single-use initiative, we've also generated a single-use design tool, which we also market to our clients, which helps them undertake single-use project design and execution. With regards to facility of the future, amongst other things, we generate concept videos to highlight facility design aspects and technologies we believe may be incorporated into facility designs in the near future. And by way of example, I just want to show you briefly our latest video, which is on a cell therapy facility. Now, as you're probably aware, cell therapy processing technologies are still very much in development phase. So we've focused on the significant logistics aspects which affect this type of facility. So I'm just going to show my screen now with hopefully the right one, and you will see the video. So this shows an autonomous truck delivering materials to a manufacturing building. And these materials are then taken by cobots using bulk scanning scanners to an automated warehouse. When required, the materials are again taken by the automatic system using cobots and bulk scanning to the production area. This shows an automatic surface sanitization as the materials pass to a cleaner area. And here you see a cobot picking the pallet, getting a container and moving it into the kitting area. Here you see another robot prepare batch crates with various components from the picked containers. And there you have the batch crates now being taken either to whip storage or the production area. And here you have an integrated logistics system which takes it to whip or to the production area. And this shows delivery to production via an automatic sanitizing airlock. And here we have a product sample being taken from production, again by the logistics system, to the QC lab, where it's retrieved by a robot and delivered to a QC stack robot system, which interfaces with the analytical equipment. And that's our vision of how a autologous cell therapy facility may be developed in the coming years to overcome the significant logistic aspects that those facilities present. So that was a very brief overview of PM Group's approach to pharma innovation, and I hope it gave you some perspective that PM Group drives innovation in this field. So once again, I'd like to give my congratulations to all the finalists, and I wish them best of luck in their future endeavors. And with that, I'm going to hand back to Matt.
Okay, thank you very much, Peter and Austin, and thank you, of course, to PM Group, our award sponsor. So before we move on, just to let you know, we're also live tweeting throughout all of our 2020 IChemE Global Awards webinars. You can get involved using the hashtag IChemE Awards. But without further ado, here are our finalists. So we have the Activated Research Company, Biosimulitics, GSK, a joint entry by North Carolina State University, Boston University and Kraton Corporation, and a joint entry from Novo Nordisk Denmark, Aldalf SPA and Novo Nordisk PARS. All our finalists have been invited to join us today and give a short presentation about their work, and subject to time, we'll be inviting you to participate in Q&A at the end of each presentation. You can ask your questions via the questions box on the GoToWebinar portal. With that in mind, please welcome our first speaker, representing the Activated Research Company. It's Andrew Jones.
All right. Good morning, everybody. Just want to make sure you can hear me all right.
We can hear you. You just need to change your display settings, Andrew, so we can see the other view. Perfect. Thank you.
Yep. Did that work? All right. Perfect. Well, as I said, good morning, and I apologize. I've been having some network difficulties, so you'll have to bear with me if there's a pause. Just let me know if there is an issue hearing me. But yeah, thank you for the opportunity to present. As mentioned, my name is Andrew Jones, and I'm the CEO and co-founder of Activated Research Company. There is a problem with the way drugs are being developed today. Now, this problem is uncertainty, which has led to wasted time, wasted money, and less effective research. And we've heard it directly from the scientists doing drug development. They cannot give you an accurate assessment of purity until it is too late. So in the process of manufacturing drugs, there are always byproducts or chemicals that come along for the ride. And in early drug development, scientists estimate the amount of these byproducts using chemical analysis. The problem is that the detectors used are very bad at estimating amounts, and they can be off by up to a factor of 1,000,000. So when scientists estimate the concentration to be within a safe limit, they may find out later in the development process, once the drug has been scaled up, that the actual concentration of the byproduct is way above the safe limit. And this seems to be what happened with Zantac and other NDMA containing drugs that were recently recalled by the FDA. As you can see here, if we estimate something below the safe limit, but in actuality it's above, we find that out later, the holy grail would be a detector that gives an estimate of every single byproduct early in the drug development process. So in short, we have these wonderful scientists who have made these wonderful new drug candidates, but they are forced to choose between a myriad of detectors that will each give them a different value for purity. It may be 95% pure by NMR, but 90% pure by mass spec. Which one is correct? The answer is none of them, because they are all estimates and not measuring actual values. So what if we could design a detector that measures chemicals and impurities accurately with a universal response that is simple and robust? Well, we consistently hear from scientists working in pharma that a universal detector would change the paradigm of how drug development is done. So I'm proud to present the world's first carbon selective detector for HPLC, which solves this problem. It utilizes laser-based photocatalysis and flame ionization technology to give a uniform response to non-volatile analytes. Now, the Solvair technology is the latest addition to ARC's analytical product lineup, and it's built on a platform of catalytic reactor innovation. Our products have been helping scientists simplify their analyses for over 5 years, and in some cases have made previously impossible analyses possible. So what all this means in practice is that whether scientists are looking at sugars, like table sugar, or proteins, they can now get a similar linear response for those and any other non-volatile compounds. And what this means for drug development is a greater amount of certainty, leading to faster drug development cycles, less waste, and ultimately, hopefully, better, safer drugs. Activator Research Company, or ARC for short, is a small product development company founded in 2014 with a mission to make chemical analysis easier for scientists. We're located in Eden Prairie, Minnesota, and we are constantly working on innovating in the world of catalysis. I'm excited to share this latest innovation, the Solvair, with you guys, and please keep ARC in mind for your future analysis needs. With that, I'd like to thank everyone for their attention and especially thank the Institute of Chemical Engineers for this wonderful opportunity. Thank you.
Okay, thank you, Andrew. We'll now pause for questions. So if you've got a question for Andrew, please type it into the questions box on GoToWebinar. And while we give you a moment on that, let me remind you that this is one of a series of IChemE Global Award webinars taking place throughout November. We do wrap up this Thursday, but there are still plenty more to come. All the webinars are free to attend and open to all. You can head to our website, icheme.org/global awards to see what's still to come. Right, Andrew, we don't appear to have any questions, so thank you very much. We're going to move on to our next finalist. Now, please welcome from Biosimulytics, Niall English.
Thank you. So I'm here to speak on behalf of Biosimilitics, and I shall enter full screen mode. So Biosimilitics is a startup company that has emanated from University College Dublin, and my name is Niall English. I'm a professor at UCD and also the CEO and main founder of Biosimilitics. And I'm going to talk to you today about our efforts to have higher quality drug design and drug development using powerful methods in searching and artificial intelligence and machine learning. So as we know, drug molecules and API molecules are developed, they're patented and so forth in their crystal solid state. That is their particular polymorph form in how the molecules are arranged amongst each other. And these forms sometimes end up in awkward Houdini-like contortions that would be very different for how molecules are arranged in the gas phase or in the liquid state, indeed. So, of course, there's a full plethora, there's a whole slew of different crystal structures and polymorphs that may well be present, and that indeed is the polymorph of the phenomenon of polymorphism. And of course, the thermodynamically stable one at a given temperature and pressure, including its storage conditions as well as manufacturing conditions, may well be quite different from the one that has actually been made that could have been kinetically stable. And this conversion to this most thermodynamically stable one could happen over minutes, days, years, decades, who knows. So finding the correct polymorph, the most energetically and thermodynamically stable one, is like the proverbial needle in a haystack. It's slow, uncertain, very costly. And getting it wrong can be very damaging. And we've all seen newspaper headlines in the past 20 odd years about various disaster stories. For example, ritonavir has many polymorph forms, and Abbott lost $250 million in terms of lost time to market in the late 90s because of that. And of course, this ends up in court as well in terms of FDA filings. And of course, there's many other examples apart from Abbott's tale of woe in the late 90s. Ranging from documented ones from 70s, 80s onwards, right up to today. For example, Tegretol, Norvir, which I spoke about, Coumadin, Vermox, Nuprobe, Avalide, and we could carry on for most of the afternoon talking about horror stories, but let's not. Let's talk about something, I suppose, more constructive or positive. Of course, the FDA and regulators have naturally taken an interest in this and stepped in, and there's much tighter restrictions now when filing for an IMPD license, as well as patents and so forth, and regulatory filings, because these now would apply to the polymorphs that have been generated, and that needs to be known and specified. So, of course, not only for manufacturing per se, but this also lends a lot of opportunities to patent specification, even patent busting and patent fighting and litigation. And the FDA are taking quite robust positions on this in recent court findings as well. So really, being able to predict crystal structures and knowing their role in the value chain as we develop pharmaceuticals is very important. And can we really, in the drug design stage, really have an exhaustive knowledge of the polymorph landscape? So that's a very important question. So as we go from the typical journey from a candidate drug initially found in a lab that might be promising to something that might be patented and then manufactured, as we all know, that could easily be 10, 12, 14 years until it would pass clinical trials and be available to the market. And of course, there's so many different molecules that could be useful that we really need to zone in on a short list of the most stable polymorphs, kinetically stable and ultimately thermodynamically stable, and really have that materials and polymorph characterization to match with pharmacokinetic profiling and so forth. So really the most we can do in terms of having better predictive power of the polymorph landscape is really essential to drive a faster, more effective and leaner, more efficient drug discovery and development process, and to get those savings in both time and money, and getting it to patients faster, which is ultimately the end goal after all. At the moment, in terms of getting a good polymorph population characterization, all we really have is experiment, typically in the guise of powder X-ray diffraction. And this is painstaking. It can take many weeks, typically many months of valuable time. But in the end, we're always left scratching our head, wondering, have we found the truly thermodynamically stable polymorph? This is one of the great questions. It's akin to a holy grail, I suppose. So we have decided at Biosimilitics to grab the bull by both horns and to tackle the two big problems in doing this virtually. That is searching the large polymorph space, the search problem, and having a better, higher fidelity, higher quality, higher accuracy representation of the intermolecular interactions that arise when you have awkward polymorph poses and molecular poses that are bent into Houdini-like contortions that we wouldn't expect in the liquid or in the gas state. Now, to do this at Biosimilitics, we have developed a suite of integrated tools powered by machine learning and artificial intelligence. We can take a simple two-dimensional sketch of any molecule sketched on a piece of paper or a computer screen or indeed a beer mat, and we can put that into, number one, our search algorithm, which will search exhaustively, and number two, we can tune the best, highest accuracy force fields from artificial intelligence. So we combine different pieces of a jigsaw, if you will, that all interconnect, and we've encoded this in a recent patent submission that's now at the European Patent Office at PCT stage. Essentially, what we output is a detailed profile of all of the polymorphs ranked by their energetic stability, but we can also, of course, print out diagnostic, such as the simulated X-ray diffraction pattern that we can then mix up with spectral matching with observed powder X-ray diffractions and so forth. So we can use a pragmatic spectral matching, if you like, or we can rank them by the most stable in terms of thermodynamic energy and free energy, indeed. And in the search algorithm, we can search very efficiently. Instead of jumping up and down through complex energy landscapes, up and down mountains, valleys, and down into crevices, we effectively remove all of the barriers by having a series of flat interconnected steps, rather like the Giant's Causeway. And imagine jumping easily from step to step in the Giant's Causeway in several dimensions on a pogo stick, and we can do that very easily. So typically, without our Biosimilitics crystal structure prediction suite, in other words, a traditional hard way X-ray diffraction, this might easily take 12 to 16 weeks. It might take 6 months, perhaps, to do a good quality polymorph screening for an IMPD filing or for patent submission and so forth. We can really reduce that time substantially down to far fewer weeks. We can get a saving of a quarter or indeed half, but we can achieve what we really want to achieve, which is a much greater level of certainty, essentially full certainty, within a substantially less time in terms of a ranking of polymorphs. Some of the challenges, of course, that we've faced have been dealing with molecular flexibility, floppy pharmaceutical molecules, of course, chirality. Many leading pharmaceuticals and APIs are chiral indeed. So our neural network that we have developed can indeed handle this very well. It's very adept, and we have machine learning encoded in what we call a PIN, a permutation invariant or independent neural network. We can predict quite well for racemic and chiral molecules and non-chiral molecules, indeed, the energy and density according to experiment. I'm just showing you a couple of molecules here from a client of ours in pharmaceutical company. And then we can predict very well the space group of the molecules and the lattice parameters and so forth, the bond angles. And of course, we can simulate very easily the X-ray diffractions patterns and match those up with experiments. So in summary, at Biosimilitics, in our breakthrough pharma technology, a little history I've shown you here. We started out in early 2018 from an Enterprise Ireland grant, for which we're very grateful, a commercialization fund. That ended earlier this year, this past spring. We've got some very good results for polymorph predictions on standard test molecules. We've published that on water ice, as well as details of our algorithms, independent of our patent as well. And we're also working on publishing details on mannitol, benzene, the Cambridge Crystallographic Data Centre's blind test for so-called molecule 22. We've done very well there. We're getting substantial commercial validation from various partners and various end license customers. We've had our first contract win, and we're also working with setting up licensing agreements with European contract manufacturers, and we are looking to convert those early next year. We've also just finished our first investment round in the past few days, and I have to thank Enterprise Ireland and some private equity investors for that, as well as we power ahead in the next 12 to 18 months with further commercial validation ahead of a Class A round or Series A round, probably in early to mid 2022. And with that, I would like to finish my presentation. I thank the IChemE very much, and also, of course, Project Management for sponsoring this category session. And I was very pleased to see many of PM's interesting activities in their presentation. So with that, I thank you for your attention, and if there's time, I'll happily answer any questions. Thank you.
Okay, thank you very much, Niall. So once again, we do pause for questions. So if you've got one, please type it into the questions box on the GoToWebinar portal. And again, while we give you a moment on that, it's a good time to thank our volunteer judging panel, led by our head judge, Keith Batchelor. The judges work tirelessly to review and score every entry across all of our categories. You can find out more about their work and our judging panel on the IChemE Global Awards web pages. Okay, we don't appear to have any questions, Niall, so we're going to move on. Thank you very much for your presentation. Next up, it's our third finalist from GSK. Please welcome Tobias Cleaver-Ross.
Hello all, can you all see my screen?
Yes, we can, yes.
Cool. Oh, just move that out of the way.
But we can't see you, we can just see a black square in your camera.
Strange, I'll change that. That better?
Perfect. Yes,
cool.
Thank you very much.
No problem. Hi everyone, so I'm Tobias. I'm a platform engineer based at GSK in a town just north of England, just north of London called Ware. And today I'm going to talk you through the batch flow optimization system, which is all around being smart instead of steel. So just to provide a bit of context and understanding, so I'm going to go into a bit of the background, then I'm going to go into the why and then the how it was implemented as well, and then going to go into a live view. I'm a firm believer of actually letting people see it and see the tools that we use. And then also then talk about how it has actually helped us win for patients, and then that societal benefit, and then time for any questions at the end. So just to give a bit of context and understanding into it, it's a system of interconnected dashboards, providing a single source of truth. Now that last bit is very key, and I'll come on to that bit a bit later on in the presentation. But ultimately, it has three main parts. So it has a batch flow system, an inventory management system, and engineering maintenance and spares part of the system as well. They all work in conjunction to enable us to reliably deliver critical medicines to patients at the end of the supply chain, reduce environmental impact due to landfill reduction and the cost of storing great inventory quantities in controlled conditions. So, but ultimately, as I said, it comes down to the patients themselves. So a bit about the background. The system itself is live across 12 sites within our pharma supply chain network. The reason why I say plus is actually now 14. So there's 2 that have now implemented it since, with a further 2 also now implementing it as well. So in total, roughly 16. They're mainly spread across Europe, with the site I work at here in Ware. But then we also have Zebulon in the US, Baronia in Australia, Emaichi Japan, and West Wharf in Pakistan as well. So what this has done is it's allowed full visibility of batch and inventory movement between the sites, providing real-time movement of medicines to packaging sites and patients. But it's also allowed all of our sister sites working together. So now we can actually share raw materials and components by seeing availability at sister sites, and if required, shift production to accommodate that demand. thus then reducing transportation costs and any further logistical costs or environmental impact from those logistical additional costs that we then have to implement. So why? So I work at a site based in Ware, England, which is actually only a small part of GSK as a whole network around the globe. But impact-wise is very significant, as we as a site operate both as a global supply and new product introduction site. So we transfer all the new products to our sister sites to then manufacture around the world. One of my previous roles as a shift manager, I would actually have to update whiteboards with laminated cards of where batches were, what spares we had available, and the cost of inventory at any one time. The problem with that is it's a very manually intensive task, as you can probably see by the boards there. The card could probably would take forever to actually update, and I would alone spend about 2 hours a day updating the boards with the correct information, which after a few hours then became outdated, because again, coming from our data system to then update it, I would then have to do it again. I came to realize that this wasn't just an isolated occurrence, but happened all over the site and consequently all over the company. It also depended on multiple departments to attend key daily meetings and update with the relevant information, bringing me back to the point earlier around the single source of truth. You would have departments coming arguing about who actually owned the batch. Was it in labs? Was it in quality? Did production still have the samples? No, yes, they were taken over to the labs. And it ended up being a clash of information at a meeting where it then defeated the point of trying to discuss where our inventory was, where our cycle time was at, what spares we had available in terms of engineering maintenance, and was the maintenance complete, and then also physically where the bottlenecks in inventory were. And just to give a bit of a flavor, this is actually just one of our products and all the different routes it could actually go through. But to put that in perspective, we have 14 alone on this and one of the value streams we have here on site. So that times 14 is the type of complexity we're trying to deal with. So how? So traditionally, pharma has been very slow in the uptake of new technologies, and it's a very manually intensive industry, or has been traditionally. And being heavily regulated, it's very difficult to implement new systems or strategies. So changing the system which people have been used to for years would be an effort. So using a combination of Power Apps with Power BI, integrated with our SAP data system, and partnering with key departments, especially people who weren't afraid to disrupt the norm, the batch flow system was then born. So that then provides a visualization of batch flow, cycle time and inventory, at-risk stock, engineering, maintenance and spares in an integrated functional system. All aligned to the common goal of improving flow of batches throughout the supply chain. So far, it's provided over £13 million in savings of environmental and business costs. More importantly, it's allowed us to deliver our critical medicines more sustainably and reliably to the patients at the end of our supply chain. So that photo there in the middle was actually where the old one by the board used to be, then replaced by a digital touchscreen with the tracker on itself to allow the departments to then interact with it on a daily basis. So I'm now going to go into that actual live view of it and go through it in turn. Just a bit of a disclaimer there, batch information has been modified just to protect our patients' safety in terms of if they do receive those batches, then obviously we want to keep them safe as the #1 priority. So just a bit of a flavor, as I said, it uses Power BI. I'm going to start with the oral solid dose tracker. Essentially, this is then looking at an end-to-end supply chain view within our oral solid dose value stream here at Ware. It tells us what batches are green, what batches are then late, or if there's any troublesome batches as well, and any issues that then occur with that. Could be documentation errors to any other issues that may occur. If I then zoom in, we can then see in a bit more detail on the cards themselves. There's a bit of color differences between them all. So we then have the actual process order number. If it's then highlighted, that could, as an example, that one's then due to ship in this week's shipping schedule. If it was then green, it could then be the week after and so on. So there is a color coordinated system. Dates of when it's due to ship, of also then when it was due for an inspection to finish. But then also in the top right here is the actual cycle time of the batch itself, and how long it's been going through the supply chain from day one, but also it's color coded. So you'll see some of them are green down here, some of them are red. And what that is a predictive side. So it's looking at, based on that product and based on the issue it's got itself into, predictively, is it going to meet the cycle time that we need it to ship to the customer? In this case, these three are no. So actually, this one is a yes. Can we then substitute that with a different batch number to then go to our patients the answer could then be yes, okay, then let's ship that one first and then save that one for a later date, ensuring that we're then delivering to our patients at the end of the supply chain. I can then click on a batch. As you can see, it then highlights it. If I zoom out slightly, you can then see at the top, it then tells you exactly the start and finish dates of every single stage it's then been through, as well as how long it's then been sat in that use decision section of 12 days, and then also when it was due to finish, 10th of November, hence it is then late in this case. You can then see cycle time in an updated basis as well, and then a slight variation for a respiratory version, which then looks at inventory that's been released from the previous stage, how long it's been sat there, to making sure that we're using that in a first in, first out lean principle throughout the tracking process, as well as then the work in progress batches, how long they have been sat there. We can then track our maintenance on a day-to-day basis as well, making sure that then each of these are aligned to a technician to then complete, as well as what our schedule complete percentages for the week, and then what specific routines we have outstanding and the areas for that. These are then get automatically assigned to a technician to then carry out the maintenance and work. And as you can see, it goes green automatically when the maintenance is then complete. And then here, finally, we then have a look at our inventory side of things as a site, which looks as an overall summary dashboard, simple red and green. Are we winning? Yes or no? So we're currently on target for our site total inventory. And then you can see across the key areas on site, in this case, exactly where that inventory is, and then where the troublesome areas could be. As I said, this then goes across all the different sites, or so far 14 sites within the business. So we ship some of our bulk tablets to IChemE, for an example. They can then see from this when they've then shipped to them, so then they don't have to worry about essentially waiting for an email from us. They can then actually then receive that and check themselves, yes, this is a goods in transit, it's going to arrive on a certain date. So that actually then is fully automated as well. So we've gone from a completely manual process to one that is now completely fully automated, doesn't require any manual intervention, and then comes direct from our SAP data system and actually intelligently tells us as users exactly where batches are, where bottlenecks are, where our spares are in terms of maintenance as well for the week, and then able to plan accurately to push those batches through the supply chain. So coming on to how it actually helps us win for patients and society. So as I've mentioned, we've had a cost of waste reduction. So we've actually had a 96% reduction in batches we then write off. So we actually have expiry dates at key stages for our batches in terms of blend hold times to ensure that we don't exceed those. These trackers then tell us that it's near that date that it needs to be next. And it allows us to then make sure that batch gets used first before being written off. We then have cost of storage material, so storing it under controlled conditions and then venting that to atmosphere. We then do less of that now because we don't have to keep as much inventory within our supply chain because we've now reduced our cycle time and we're able to then meet that more reliably as well. As I said there, the increased delivery of medicine to patients, so we're able to deliver that on a more reliable and sustainable basis. But also developing digital skills within the industry. So one thing is that no one really used Power BI or Power Apps before this. When it then came about, people were then intentionally using a tool, in this case Power BI and Power Apps, without realizing they were. So then they're able to then take those skills and then apply them to their areas and then create their own different types of trackers, maybe for lab samples or for test adherence or, for example, overall site engineering maintenance. They're able to then take those skills and then use them themselves. So it was actually that change management piece, actually getting people to use the tools without realizing they're learning a new digital skill. But also data accessible to all. So now with that SAP data that I then pull into my database, I've made that accessible to everybody within GSK. No charge, completely free, and it was all done as a project in terms of it was done in zero cost, all done using Power BI, but that data accessible to all is quite a key point. It used to be very, very difficult to extract our own data. We can now do that very simply and easily, saving hundreds of hours of wasted time that people would normally do per week to allow them to then hook up to their own systems. I'd like to thank everyone for listening. Thank you very much to the IChemE judges and PM group as well for sponsoring this award. If you have any questions, thank you very much.
Okay, thank you very much. We'll give you a moment to write those questions into the questions box. We have our first question. Are you intending to proceed to e-batches and release by exception?
So we are actually looking at electronic batch records, yes. However, no, we will still have that certification done by a QP at the end of the supply chain, although it's something I would definitely be keen to look into.
Okay, thank you very much. We don't appear to have any more questions, so we will move on. Representing the joint entry from Novo Nordisk Denmark, Adalf SPA and Novo Nordisk PARS, it's Peter Fairhurst.
Right, good afternoon, everyone. Can you hear me okay?
Yes, we can.
Can you see my presentation?
And yes, we can see your presentation. Thank you.
Right, I'll just put into presentation mode. Right, yeah, so again, as the others have said, thank you very much for giving the opportunity to present this project. My main motivation for submitting this entry to the IChemE was to, in the pharma category, was to highlight the importance of medical devices and the importance and complexity of their manufacture within the pharma industry, and how good chemical engineering base can allow you to lead such projects.
Peter, sorry to interrupt. We've got the wrong PowerPoint view. If you could just switch your PowerPoint to presentation view rather than presenter view, that would be great.
Okay, so let's have a look.
You're testing my French here, but it's the top left, the second item across. Should change it, that drop down. The next one along.
Oh, sorry, paramount. Yeah, sorry. Yes. Okay, good. Is that looking any better?
Perfect. Thank you.
Good. Thank you for stopping me. And if I'm making other presentation mistakes, just let me know. Yeah. So, talking about the importance of the medical devices, if you actually look in value to the end patient, only between 30 to 50% of prescriptions are followed as they should be by the end patient. So anything that we can do, whether it's in better instructions for use, more easy to use medical devices to improve that loss of value within the in the value chain, is just so important. And that's where the good, easy to use medical devices really comes in. So if I now just move on my presentation, so you can see I've got a couple of images on the first slide. So I should just also mention, so it's a joint entry between the main Novo Nordisk company, which is a Danish company based in Denmark, and then two affiliates, Aldaf SPA, which actually stands for Algeria Denmark Pharmaceutical Company, which is the affiliate of Novo Nordisk in Algeria, and the Iranian or Persian affiliate based out of Tehran, with this head office is there. The two photos I'm showing here are photos of the two sites. The one on the top left is one coming from the Algerian site. They're both copies of each other. So the layout, the equipment, whether it's the administration offices, the production facility, or the cold and ambient warehouses are the same. So you have a sort of a side view, and here you can see we've actually got the nice green grass from the automatic watering there. And then the bottom photo is the photo of the Iranian site. And there you can see it's sort of in a stepwise configuration, where the lowest step is the administration offices. The middle part is the production area. So that's the part that I designed, and we've installed all the production equipment in, and then the back part is the logistics area with the docking stations and the ambient and cold storage. Now let's yes. So I should have moved on now to the next slide. Is that clear? Yes, good. So the content of the presentation today is just a quick introduction to Novo Nordisk and myself, then an overall description of the project. Then I'll touch on a number of the aspects of engineering excellence which have within the framework of this project, and then a little bit about the some of the numerous, let's say, project challenges that we had. And yes, again, as I go through, I'll just mention the different images and photos that I'm showing. So this is an image of the assembly area in the Iranian site. This gives you an idea of some of the production areas. So you saw the outside a moment ago, and now you're seeing the inside. So for almost 100 years now, Novo Nordisk has been translating the unmet medical needs of people living with serious chronic disease into innovative medicines and delivery systems like our insulin pens. Our treatments today are benefiting millions of people living with diabetes, obesity, rare blood and endocrine diseases worldwide. So we have a number of famous users of our products. So in terms of our human growth hormones, Lionel Messi has used those to get a bit bigger when he was a young boy. And in terms of our insulins, well, Theresa May is a user of our flex pens and our insulins, for example. Some small facts and figures. So Novo Nordisk produces about 50% of the world's insulin, and is the world leader in insulin production. About 30 million people use Novo Nordisk diabetes care products on a daily basis. And the Novo Nordisk Flex Pen, in terms of if we just talk medical devices here, is the most widely selling prefilled device ever in the pharmaceutical industry. Nowadays, and over the last 5-10 years, over 1 million Flex Pen are sold every day worldwide. So that's more than 700 a minute. Just to give you an idea of the sort of volumes that we are producing and delivering to the market and to our diabetics. And basically, the Flex Pen is an amazing success story. It was the first pre-fill pen which was delivered to the market. It managed to overcome, by its simplicity and its form, a lot of the social stigma which surrounded diabetics with a syringe and a vial and injecting those products into them. With the developments of the pen, the thickness of the needle has gone down to very few micrometers now, so the comfort of injection is much better, and the ease of injection in terms of dosing is much easier as well. So it's really a huge success story for the pharma industry and for diabetics. Very much a win-win story. Just a couple of things about me and why I'm into this. So I came from a chemical engineering degree at Cambridge University. I then did an international European PhD, actually in food engineering, a lot to do with fluid dynamics, in fact. And then I actually haven't been back to the UK since. So I've had basically an international career ever since, working in Switzerland, Canada, and I've been back in France now for the last 15 years with Novo Nordisk, but mainly working on international projects for the head office rather than for our local production site in France, in Chartres. Now a little bit of background onto the project and why we were building these production facilities for flex pens in Algeria and in Iran. Well, the first thing is the sort of emerging market trends in Asia and Africa. Growth in diabetes is often double digits in Africa and Asia. If I talk about Algeria in a little bit more detail, with the sort of the cultural norms that exist, not a lot of sport is done when people get to 30, 40 years old, so that doesn't help. Food is readily available, and then on top of this, especially for the ladies, unfortunately, when they get married, they're less allowed out of their house on their own. So they get to a phase of getting a little bit depressed and sitting at home and eating more. So in fact, we have a higher level of obesity in young ladies, for example, in Algeria, than we have in Iran. So you have this very high level of or high growth of obesity in a relatively young population, as it were. On top of this, if you look at the economic situation, both Iran, Algeria, and many of the other countries in these zones, they have really excellent education systems, delivering really well-trained, intelligent people onto the job market. But at the same time, there are very high levels of unemployment. So putting these two together, more and more we are seeing from a Novo Nordisk perspective, the governments are increasingly requesting that production of vital pharmaceuticals becomes local, for us to maintain market access to those markets, and specifically in these cases, Iran and Algeria. So that's sort of the business driver of these projects. Please become local, otherwise access for your products to the diabetics who are using them will become more and more difficult. The photo here that I put into the presentation, that's actually as I'm coming into land in Alger in Algeria. I put this one in because quite often, a lot of the Danes at least, believe that you sort of land in the middle of sand dunes everywhere, and that's just not the case. It's very green, Algeria in the northern sort of 100 kilometers close to the Mediterranean and the various mountains that they have. So then the project, as I've said just a little bit, was to establish this local manufacturing of FlexPen in production in Iran, Algeria. These were basically greenfield sites. Well, I say greenfield in the terminology, as you can see, it's basically a dirt field, which we started with. And then through construction of these manufacturing sites, diabetics will be able to continue to access their preferred diabetes care solution, thus allowing optimal management of their diabetes and avoiding all the typical complications you have when you don't treat your diabetes well. So that's loss of limbs, loss of eyesight, increasing cardiovascular diseases, etc., etc. And then on an economic side, these at project completion, so obviously there's an awful lot of employment which is generated during the project phase, but at project completion, we have about 150, or we do have about 150 employees generated directly on the site, and then with the indirect jobs which are generated is about 600 in total. So it's not going to change the world in these countries in terms of their high levels of unemployment, but it is a good step in the right direction. And here, this is a photo from the startup of the site in Algeria, and you have the Shirar Mountains just behind, which were great for running in the evenings, I can tell you. Very beautiful place. On then to engineering excellence. Well, just a little bit more details on the FlexPen, and a little bit more about the challenges of assembling hundreds of these continuously with absolutely no quality issues. So a FlexPen comprises of 11 plastic components, and it's assembled on a automatic assembly line which comprises of 96 different stations. We have different colors and for different products and different plastics and plastic compounds which are used. The FlexPen itself contains a cartridge of 3 milliliters of insulin, so it doses 300 units of insulin. It has a maximum unit dose of 60 units and a minimum of 1. If you're looking then at risk analyses, the process failure mode and effects analysis for the assembly line its own is almost 2,000 lines long. So there's a huge number of analysis and risks of which needs to be taken into consideration. The component failure mode and effects analysis, which is sort of its brother or sister, is similarly in length. So every time that, especially in these projects where we're having to change a lot of the sub-assemblies when we're going into Iran on the equipment due to the US embargo restrictions, all the analysis which had to really be done to make sure that in terms of a process failure mode and effects analysis, there were no problems, was very long and very lengthy, and you then had to take into account all of these changes into your validation setups and your validation protocols. The lines that we make are producing about 200 pens a minute, and as mentioned, all must function correctly, otherwise you have potentially lethal consequences for your diabetics and your clients. And of course, that is just totally unacceptable. And then within this as well, we were looking at trying to make the configuration management optimized. So we're typically talking here about your configuration item list or critical item list of your parameters. So as mentioned, you know, it's a very complex device to assemble with extremely as in all the pharma industry, quality levels to achieve. Continuing on then with the engineering excellence, a little bit, as I mentioned, these were greenfield sites. So I had actually I was one of the best projects I've ever had the pleasure of running. I drew with the team the layout for the site for the production part of the site, and then 2 years later I was walking around the site showing people why it was like this and why why this was optimized. So a few elements. First of all, if you're looking at in environmental health and safety, when we're looking at these new lines, we're quite lucky within Novo Nordisk that we have an EHS portal, which has all the incidents and near misses and accidents that have happened on production lines over the last 10-15 years. So when we're in the design phase, we reviewed all of the relevant incidents and then made sure that we had put into the design necessary corrective actions. Especially now in Novo Nordisk, there's a huge focus on ergonomics, and so we also had to take a lot of make a number of modifications to the line to respect the new ergonomic requirements. So, for example, this is no lifts above 15 kilograms, no lifts above 5 kilograms, with no repetitive lifts more than 5 kilograms, rotation of the bust, these kind of things. And this was actually an interesting challenge within the project, because Novo Nordisk EHS requirements were far higher than what you would find typically in these countries. We were targeting to have, in terms of noise coming from the lines, lower than 72 decibels. In the EHS requirements in these countries, 80 decibels was fine. I mentioned these lift requirements that we were looking at, up to 80 kilograms was fine in Algerian EHS requirements. We then also incorporated into our validation protocols the latest risk-based assessment approach, which allowed us to lean down by about 40% the validation time required. And as mentioned, we managed to incorporate multiple process improvements into the revised designs. So overall, a great step forward in terms of our processes and equipment, a great step forward in our validation principles in terms of concentrating really on the critical areas, all done in challenging environments, being in these new countries and having to do a lot of the work at a distance. So I'm just looking at my notes of what I wanted to say. Is there anything else I wanted to mention here? Yeah, I guess that's about it. Then just mentioning on this photo, this is a photo of the quality control lab, and we have two of our QC technicians here from Algeria who are performing some dose accuracy tests of the prefilled devices, of the flex pens. And again, here, this is a good example, a little bit on the ergonomics. So they're sitting at tables where they can adjust the height, so it's ideal for their height. We have these vacuum extractors to remove insulin fumes so that they don't inhale them. And then you also can see some additional lights which are put in to ensure they have optimal lighting. And all of this is done on a vibration-free floor, so that we have the necessary accuracy and reproducibility and repeatability in the QC measurements. On to a little bit of the project challenges. So I have 2 slides on project challenges. One is on the common project challenges across both projects, and then a slide talking about specific challenges in each of the countries. And first of all, here, the photo here is the Algerian team after they finished their last packaging process validation batch. It's actually 2 o'clock in the morning, not 2 o'clock in the afternoon. This was done just as the COVID pandemic was hitting us, and we were in belief that there would be a global lockdown in Algeria and nobody would be allowed onto site the following day. In actual fact, that shutdown didn't happen the next day, but it happened 2 days later. So there was this huge push to finish the process validation batches before that happened. And the reason why they're all holding up their hands with 4 fingers up is because it was the 4th batch. We're doing 4 validation batches in this particular case. And as you can see, they're looking pretty happy. So yes, one of the first major challenges here was in both of these countries were very unsighted in the duration between submission to the local regulatory authorities and then the approval to manufacture to market. In the benchmarking studies of other products which are being submitted, this could take between 3 months and 2 years. And well, sort of obviously, if you've got a 2-year wait between when you've done your process verification batches and when you can actually produce to market, or 3 months, is quite different of how you are going to sort of manage your recruitment strategy or manning strategy around that. So it was a big challenge within this project of trying to work out when to recruit locally, how much to recruit locally, and then how you would retain and motivate locals who you had recruited locally if and when you had these longer durations between submission for the right to produce to market and when you could actually start producing. The second big challenge, which led to an awful lot of discussions with regulatory authorities, was that the Iranian FDA, like the Algerian FDA, they're used to packaging pharmaceutical facilities and they're used to filling pharmaceutical facilities. But when you come with what is basically a medical device assembly site or a durable a drug device combination product, as it is also called, then they're sort of a bit lost. What validation procedures should they be looking at? What GMP requirements, etc. In the FDA, you have a complete section on medical devices, but typically in the Algerian Iranian authorities, they're not used to this, so you're having to sort of educate them at the same time as trying to present how you intend to present your files for submission. And that took a long time to get people to understand. And of course, you're into government offices where there's a flow through, there's an election, people change, you have to restart again. That means certainly in Iran, you're talking about a lot of tea drinking. Next one I put down was the management of the local environment and safety requirements. So different safety requirements, which ones do you follow, corporate ones or local ones? We're trying to be cost competitive also in these countries. So we didn't want to put a Rolls Royce into a country which isn't prepared to pay for that. So again, there I had to find the best and adapted solutions, as it were. Lots of replanning and interdependencies. Sometimes they say project managers are spending 70% of their time replanning. There were stages in this project that this was the case. Another challenge is that you're requested to source local very early on. So within 6 months, we're asking you to source all packaging materials locally, even though a lot of local suppliers cannot deliver the quality that you want. So then you're into a partnerships very early on to try to get that to work. And then we had COVID-19 hitting the project at the end, which means that even though we had these composite central local teams, then suddenly the central team coming from France and from Denmark couldn't travel. We had to then reorganize the teams, do a lot more virtual training, etc., to be able to finish the project. A little bit on the challenges on the specific countries. I think I mentioned a bit earlier. In Iran, especially when the United States pulled out of the nuclear agreement, this US embargo meant that we had to redesign our lines by 20 to 30%, which is a huge challenge, and then make sure that we could get these lines to work and to produce the same quality, etc. Again, with these kind of things, you surprise us all the time. We need a high quality vacuum for running these lines. We had our vacuum pumps stuck in Dubai for 9 months, as these vacuum pumps could be used in the nuclear industry. So we were buying off eBay or the equivalent of eBay in Tehran some second-hand vacuum pumps while we were waiting to get those ones into the country. Test materials, so as you'll all be aware, when you're starting up these lines, we're not using insulin in cartridges, we're using coloured water. the Iranian authorities could not understand this. They blocked our coloured water cartridges for 12 months. That's a challenge in terms of your project timelines, obviously. And so a lot of time, again, explaining to customs going and doing this. Novo Nordisk does not make facilitation payments, so I think this also slowed down the project quite a bit, as it were. Persian culture is very much relationship-based and not time-based. So when you're asking for timelines, you say, let's have a cup of tea. When you as a stressed European, you're saying, but when are you going to finish? And then you get told back how well they're doing, but no timelines coming back. So that's always a little challenge. In Algeria, you have very heavy and long administrative procedures for everything. From a management perspective in Algeria, you always have this very much optimistic communication of when things will be done. So it will be finished next month when you know it's going to be 8 months' time. So it's not a question of being untruthful, but it's like from an Algerian perspective, if you want something finished in 6 months, you have to target next month, as it were. And that took a while to incorporate into our project plans and our communication strategies. And then the last one I put down was while we were running this project, we had the presidential elections where Bouteflika was going to stand for a fifth mandate. I don't know if you saw that, but at one stage, for a country of 40 million inhabitants, you had over 25 million protesting. And so that caused significant issues in trying to go into the country and have access to the site and so on. So we actually had to put in cabins with beds and food, and had to have people with satellite phones if ever we couldn't leave the site because of all these government protests and so on and so forth. So not a simple life as a project manager. The state hours to both sites have been completed. All submission has been done. The Iranian site is now producing to market, and we are waiting for return from the Algerian authorities to be able to produce. There have been no technical issues raised with the submission files for Algeria. I think it's more now a question of negotiation of the price point of our various products. So that is the end of my presentation. Thank you very much for listening. And yes, I guess any questions I can take now. So I should now be off the presentation mode, right?
Yes, thank you very much. Yes, we can still see your slides, but don't worry about that. We are going to have to move on in the interest of time. So thank you for that presentation, Richard. But we do have one further finalist to present. It's representing the joint entry by North Carolina State University, Boston University and Kraton Corporation. Please welcome Richard Spontak. Okay, Richard, we can't hear you yet. So we can see your screen. We can't hear you just yet.
Can you be able to hear me now?
Yes, we can. Yes, we can.
Okay, perfect. Sorry, for some reason it was not letting me share. So first, let me begin by pointing out our deep appreciation to the selection committee of the IChemE Global Awards for choosing our work for being a finalist in this competition. That is indeed a great honor. We are most appreciative of that. This is a collaborative effort between NC State, Kraton, and Boston University. And I'm going to be showing you some of the work that deals with self-disinfecting polymers, especially as it corresponds to COVID-19 as well as other human coronavirus. My name is Rich Spontak. I'm from NC State. In terms of chemical engineering, both of my degrees from Penn State and from University of California at Berkeley are in chemical engineering. And then I have ties to Cambridge since I did my postdoc there. and then worked for Procter & Gamble for some time, where I became first exposed to antimicrobial materials, and then have continued on in trying to engineer the chemistry of materials. Professor Galati is a worldwide expert in terms of antimicrobial approaches, primarily dealing with photosensitizers. Professor Scholla is an expert in viruses. Dr. Mehtar from Kraton is the CEO of Kraton. So he has been an active participant in this work. And Professor Anthony Griffiths from Boston University, who is also a member of the National Emerging Infectious Diseases Laboratory, is an expert in infectious diseases. And so with that introduction, let me go ahead and get started. We've all grown painfully aware of seeing diagrams like this over the past few months. It has not been easy to watch this, and every time I give a presentation on this topic, I feel obliged to update it. As you can see here, I've updated it as of this morning, US time, just to keep it current. And every time I do this and I look at the numbers, it never gets old in terms of appreciating how many people have paid the price for some of the mistakes and some of the shortcomings of our healthcare industry. And so what you're seeing here now, as of today, we're looking at about 1.3 million deaths, with about 250,000 in the US alone, about 50,000 in the UK, and we're looking at about 55 million global cases. So this, as we all know, has essentially brought society to an almost standstill. And this is the culprit for this pandemic. This is a visualization of the SARS-CoV-2 virus. And the numbers I just quoted you are listed over here. And I think the one thing to always remember is that every time one uses numbers like this in a statistical approach, we need to remember that behind every single digit of these counts are people, people who have families, who have friends, who have dreams, and we cannot forget about those who have suffered through this or who have perished through this. So we want to look at how we can move ahead. And certainly the use of personal protection equipment is of paramount importance. Using a face mask, in some cases a face shield, paying attention to social distancing, those are all very important. And now we have good news on the horizon, such that we might be seeing some vaccines that really are showing a lot of encouragement. But that doesn't mean we simply stand still and not do anything. We have to ask, can we do better? And so one of the points in which we can do better is through the use of materials development, in particular, a polymer like Biaxin, because this material has truly unique properties. And I'm going to share with you some of those properties today. First, as I mentioned earlier, a lot of my own personal interests involve the development of new materials and the use of materials in different ways. And so the material that you see here and the chemistry involved in this material is truly insightful, because this is called a block polymer because it has these contiguous sequences, and there are 5 of them, so this is a pentablock. And what's nice is that the end blocks are T-butylstyrene. So these are glassy polymers. They impart strength to the material. They allow it to be made into films, or you can make these into coatings. There is an ethylene propylene rubber intermediate block, which imparts flexibility and therefore makes the material tough. So it's not going to simply break if you try to bend it. In fact, this material belongs in a very special class, which I'll mention in a moment. And then lastly, it has a styrenic mid-block, which is partially sulfonated. And because of this, it becomes both antibacterial and antiviral. Now, block polymers are known to microphase order. They self-assemble. There's been a lot of work that has gone on around the globe trying to understand and explain this kind of behavior for this class of materials. It's one of the most comprehensively studied materials in polymer science. And so we have to ask the question, is this going to be something that will be of broad use? Well, the answer is that, first of all, as I mentioned, it belongs to a class of polymers collectively called thermoplastic elastomers. And these types of thermoplastic elastomers are essentially replacements for just chemically cross-linked rubber in many cases. They exhibit a lot of similar properties because they have rubbery blocks and glassy blocks. The glassy blocks act as physical crosslinks instead of chemical crosslinks. And you can see here that globally speaking, we're looking at a $22 billion a year industry as of last year, and it's projected to come up to about $28 billion by 2024. And at the top of this list are, in fact, styrenic block polymers, of which we see one right here. It has styrene and it is a block polymer. And one of the most important companies in the world for processing block polymers as thermoplastic elastomers is, in fact, Kraton Corporation. And you can kind of get an idea here that now this type of material, which is referred to as, again, biaxium, is going to be a thermoplastic elastomer that inherently is still self-disinfecting. And the antibacterial or antiviral compound is chemically bound to the molecule. It cannot come off, which means that we now have materials that can be produced in large quantities and used in a variety of different applications, as I'll point out later. And this just is an indicator that this material is produced at commercial quantity. So it can be used right away for combating the spread of COVID-19. So as I mentioned, it's a charged block polymer, which means it has hydrophilic channels. Those hydrophilic channels that measure on the order of tens of nanometers allow the migration of protons from the sulfonic acid groups to the surface in the presence of water. So here are some 3D TEM images that show the structure of this material, and you can see that it depends on which solvent is used to cast it, but we produce a morphology that, especially in this case, you can see the channels very, very easily. And what we also find is that the surface of the material restructures in the presence of water. So without water present, we see that there is no signal from sulfur, from the sulfonic acid groups, according to X-ray photoelectron spectroscopy. But now if we put it into phosphate buffer saline solution, we not only see calcium, which is present in the solution, but we also see the emergence of these sulfur peaks, indicating that they now abide at the surface. And that is what actually winds up allowing the surface to become highly, highly acidic. How highly is highly? Well, pH levels below 1. And this is only at the surface, but what it effectively does is once the virus particle comes in contact, this highly acidic environment basically deteriorates the membrane of the virus. So as a quick overview, yes, this type of material is very effective against a wide range of microbes. In fact, we were very grateful last year when we were chosen as a finalist because this material was very, very effective against a wide range of bacteria that you see here. And now we can actually add to this list C. difficile. We looked at a number of viruses to show that this material was highly effective against viruses as well. And more recently, it's been examined against additional viruses and mold, as well as SARS-CoV-2 by our collaborators at Boston University and independently at the University of Texas Medical Facility at Galveston. And you can see the results over here that show that we're looking at very effective inactivation over very, very short times of exposure. So we see that, yes, it is capable of inactivating SARS-CoV-2 in 5 minutes. And just to give you the data, here's the survivability as a function of time. There's 5 minutes. And 10 and 15, you'll notice are all about the same. At longer times, we're looking at a higher concentration of the initial virus suspension, which is why it appears so much lower. But compare this now to copper, which is a well-established antiviral surface. And here we're looking at 1 to 4 hours based on some work that appeared in the New England Journal of Medicine and sponsored by the National Institutes of Health in the US. And you can see that, yes, there is activity, but it is far short of what we can achieve in just 5 minutes. And that is repeatedly. It's not just a one-time deal. It can last, we know that SARS-CoV-2 can last for days on various substrates, and those contaminated surfaces are referred to as fomites. And depending on the temperature, at low temperatures, that time of survival on surfaces can actually approach a month. So we wanted to study this in a little bit more detail. And because we don't have a BSL 3 or 4 lab at NC State, we used a human coronavirus surrogate, which is human coronavirus 229 echo. This particular virus is what's responsible for the common cold. And it also survives for days on various surfaces. And what you can see here is that all the materials that we've studied, including Biaxin, which we refer to here as TSAT52 because it contains 52 mole percent sulfonic acid, all of these materials that we've looked at have been very effective at killing this particular coronavirus. The one that you see that doesn't work is because it has a very low sulfonic acid content, only 17 mole percent, and so it's insufficient to actually work as a true antimicrobial material. What are the advantages of having these types of coatings? Well, they can be used in a wide number of industries. The healthcare industry, in terms of coatings for face shields or face masks, or being put on gowns to protect the wearer, or in the transportation sector for commonly touched objects, or touch screens that are more and more commonly employed for collecting tickets, or even within airplanes or trains or automobiles, so that the seats can be made antiviral, or the seat belts can be made antiviral. And then, of course, in buildings, one could imagine having tabletops that are coated with an antiviral. Or we could talk about filtration systems. And again, commonly encountered objects where people can touch in addition, after, for example, coughing or sneezing onto their hands, and then touching and depositing the virus onto these surfaces. So it can be used in a number of different ways. And one of the most important and exciting aspects of this material is that because they are not selective, they afford no possibility of resistance on the part of the microbes. So they cannot become resistant in the future. And that means that this particular approach is not only valuable against COVID-19, but we could think about COVID-21, COVID-22, and so forth. Because right now, the data suggests that it won't matter what type of strain we're dealing with. This type of material has already been commercialized for other uses, such as for cooling purposes in sporting goods or humidity management in air conditioning systems, or even for water purification. But it also shows additional applicability as solar cells, where we can achieve about 7% efficiency over a wide wavelength range. It can be used for electroactive polymers for use in robotics. It can even be used for gas separations so that we can enrich one component and remove it from a mixture of gases. So think about, in this case, removing ammonia from biogas. So it has lots of versatility. Now, it is important to realize that anionic polymers have been known to show variocidal activity before. In fact, it goes back almost 50 years, but the big difference is that all of those studies were in solution where acidic polymers were added to prevent viruses from gaining entry to cells. The polymers that I'm talking about here, in particular the Biaxum, they do not dissolve in water. They remain as solid surfaces. And so they effectively prevent virus transmission by touch. All of the anionic block polymers I showed here exhibit well-defined hydrophilic channels, and that is really very important. But we also wanted to see if this was a general approach, and the approach is general, because now if we look at a different type of amphiphilic polymer that also exhibits water channels, but these channels are much smaller, on the order of just a few nanometers, this is Nafion, which is commonly used in fuel cells. And what you see here is that because it also contains the sulfonic acid groups, it is an anionic polymer, but it's also highly fluorinated, as you can see. But you could, in terms of the human coronavirus, after 30 minutes, we get close to the minimum detection limit once again. So this approach is actually very general and can certainly aid us in future chemical engineering of materials to prevent the spread of viruses as well as other microbes. So with that, I would like to point out people who have been involved in this project. From NC State, Bharadwaja Padinti, who is pictured here because he was the one who effectively discovered this whole approach of anionic polymers. Along with Jiaqi Yan, we have our colleagues at Creighton and Boston University, and the work has been sponsored by Creighton, Halyard Health, the Nonwovens Institute at NC State, and the Research Triangle Nanotech Network. And in closing, I want to kind of give you a preview that, once again, this material is surprising because Clostridium difficile, it's not a virus. It's a gram-positive bacteria, but it is responsible for also about 250,000 deaths per year in the US alone. And that amounts to a price tag of about $1 billion. This particular microbe is difficult to kill. Here are results that we just obtained about a week ago. And you can see that in 1 minute, Biaxum can kill 99.99% of the vegetative C. diff and it takes about 60 minutes to kill 90% of the spore cells of C. diff. So it has, as I mentioned before, it may take a little bit longer in terms of exposure times, but these materials offer tremendous potential for protection in the future. With that, I'd like to thank you all for your attention. Again, we are deeply honored to be selected as a finalist in this competition. And we certainly wish all the best to all of the other finalists in the competition. Our contact information in case you would like to reach any of us. And as a final note, please wear a face mask to stay safe, not just for you, but also for those who are dear to you. Let us remember to stop the spread. No, let's do one better than that. Let's prevent the spread. And with that, thank you very much for your attention.
Thank you very much, Richard. So once again, it's time for Q&A. So if you have a question, please type it into the questions box. We have our first question. With such a low surface pH, won't human skin get acid burns?
That is a question we see very often. And the answer is absolutely not. Because it is only at the surface, it's only within the first, we haven't measured precisely how deep that layer is, but we are estimating it to be on the order of tens of nanometers. You would never, ever feel that. And in addition to that, the outer epidermal layer is already dead, so you would not feel it. Now, if you put your tongue on it, that's a different story. But I'm not advocating that one go around licking these materials. But clearly, if we're talking about just finger touching and so forth, no, there would be no issue of that whatsoever. And in fact, we've already done that experiment.
Okay, thank you very much. That's the only question we have. So that brings the finalist presentations to a close. And before we move on, I'd like to thank all of our finalists for being with us today and presenting on their work. I'm sure you'll agree, a range of very good, very different presentations. So now it's time for what everybody's been waiting for, which is to reveal the winner. So we'll start with our highly commended entries. And they are the entries, the joint entry from Novo Nordisk Denmark, Adalf SPA and Novo Nordisk Pars, and the joint entry by North Carolina State University, Boston University and Kraton Corporation. So congratulations and well done to everyone associated with those projects. But now to announce the winner, I'd like to hand back to PM Group's Peter Farrelly.
Thank you very much, Matt. Firstly, congratulations to all the finalists. There were some excellent entries and some really impressive presentations. So now to the winner. Ladies and gentlemen, the winner of the IChemE 2020 Pharma Award, sponsored by PM Group, is GSK.
Okay, congratulations then to GSK. And Tobias, if we can get you back on the line, how does it feel to be an IChemE Award winner for a second time this year?
Absolutely amazing. This is such a good feeling. Thank you very much. Thank you to all the judges. Thank you to the finalists for presenting today, and to PM Group for obviously sponsoring the award, and thank you to GSK for all their support that they've given me as well.
Well done. As I say, it's the second award for GSK this year, and of course now contender for Thursday's top prize. So well done. Big thank you to everybody who's joined us today, particularly our finalists and of course our sponsor PM Group. Don't forget the IChemE Awards webinars continue to come thick and fast this week. They're all free to attend and open to all, so hopefully you can join us for more. But until then, thank you very much for being here, and congratulations to GSK, winner of the IChemE 2020 Pharma Award.